So while this is setting up, today I'm going to talk about mostly some of the published work on the human blastoid, and actually I'm going to talk about another embryo model that's unpublished. This is the first time I've been presenting in front of audience outside of the institute. Hopefully you guys can give me feedback on what it is. Okay, so my lab started about 5.5 years ago, and mainly focused on this very unique cell type called proponent stem cells. Proponent stem cells can be derived either from an early embryo, such as a blastocyst, by culture them in a combination of growth factors and chemicals to stimulate the growth of the epiblast so that they can proliferate and self-renew in the dish as a form of embryonic stem cells. The other strategy to generate these type of cells is to use a cell reprogramming approach pioneered by Shinaka by forced expression of 4 transcription factors that are specific for the proponency, including SOX2, OCT4, and CMYC and CAL4. And these factors can induce somatic cells into becoming embryonic-like cell state called induced proponent stem cells. So while these cells are stabilized in culture, they retain these 2 remarkable properties. One of them I just mentioned, that they can produce copies of themselves over and over again in the process called self-renew. The other ability is they maintain the ability to differentiate into a variety of different cell types, tissues, and organs in our body. They call it pluripotency. So my lab is using, one of the focus of the lab is using proponent stem cells to model early human development. We're very interested in this period between 2 weeks and 4 weeks in human development, which the reason we do that is because it's essentially a black box of human development. We don't have access to the materials to study this developmental period. And so to do that, instead of using proponent stem cell, we were asking the question whether we can use this in vitro cultured cell types to recapitulate early developmental early embryo, for example, a blastocyst or later structures that are a little bit slightly later developmental stages. So there's several reasons why we do that. One of them, especially for humans, it provides accessible experimentation because, as you can imagine, it's really hard and really difficult technically and also ethically to obtain tissues, human tissues during this developmental period. And it provides a model, in vitro model or accessible model to study early human development. And also, if we can push this model a little bit further, we can potentially generate a unlimited supply of embryonic tissues, progenitor cells, or even some organ primolecules. But more importantly for me is that I'm quoting Richard Feynman's saying, what I cannot create, I do not understand. Through this process of modeling, assembling different types of stem cells together to generate structures, we can learn how human development, many of the secrets of human development, they're otherwise not able to discover. So there are many different types of human proponent stem cells have been reported, starting from 1998. The first human embryonic stem cells were derived by Jamie Thompson using yeast embryonic day 6 human blastocysts, growing them in culture that at the time we didn't really know how to culture these cells. Researchers tried to use the mouse embryonic stem cell culture conditions, including LIF and BMP, but it didn't work for humans. Ended up that the human embryonic stem cell derived at the time requires supplementation of FGF and TGF beta, which later we realized that cell culture in those conditions are more resemble post-implantation human epiblast, or now we call it prime state. This prime proponency state are more responding to this E9 to E12 epiblast instead of the epiblast from the human blastocyst. Although these ES cells initially were derived from the blastocyst, but the progress in vitro developed a little bit more, and then settled in at this prime proponency state. And also in recent years, we have been able to generate other type of human proponent stem cells, as you may heard of. Some of the cells are collectively grouped as a naive human proponent stem cells. They are cultured in the different combinations of chemicals and growth factors. So transcriptomally, they are very similar to human E6 to E7 epiblast. So they are more closer to pre-implantation human blastocysts or epiblast cells in the blastocysts. And also there are variants of various culture conditions that can stabilize what we call intermediate or according to Austin Smith, a formative state of proponency. So these cells are in between this naive and prime proponency. They are having features that are more similar to E7 to E12 human epiblast. And one of the key features, functional features of these cells in the intermediate state is that they can directly respond to BMP signalings and generate primordial germ cell-like cells in a culture dish, which the naive cells and prime cells, especially in mice, they are not able to respond. And with all these different types of stem cells, it's very important to choose the right cell type to do the embryo modeling. And first, I'm going to briefly talk about this human blastoid, how we came upon a solution to generate this type of structures. Is because of this unique feature of naive human proponent stem cells. So unlike mouse embryonic stem cells, which we consider naive, human naive proponent stem cells are not settled in their lineage, so they still retain this plasticity to generate actual embryonic cell types, such as cells that are mimicking looks like a trophoblast cells in the upper layer of the blastocyst, and also under some conditions you can generate this hypoblast-like cells, which later on will generate the yolk sac and extra embryonic membranes that support pregnancy. So this is a very unique feature of humans in contrast to mouse, because mouse embryonic stem cells, as we know, they can only give rise to embryonic tissues and also the actual embryonic endoderm, but not trophoblasts easily. You need to either knock down certain genes such as OCT4 or overexpress transcription factors that are specific for trophectoderm development, such as CDX2, in order to convert the mouse cells into those actual embryonic lineages. But in human, you can simply do this by culture adaptations under different signaling inhibitions and activations. And this plasticity is reflected in the early human development as Austin Smith's group showed that if you take out the human epiblast from a human embryo, and under certain conditions, those epiblast cells can give rise to trophoblast cells in the culture dish. So this is very different between human and mouse. They are more plastic in the human development early on. So harness this bipotential or the plasticity of this naive human proponent stem cells. Two of my postdocs, Le Qianyu and Yulei Wei, at the time, they were a couple, they came to me and said, Jun, maybe we can develop a protocol that can generate blastocyst-like structures using naive proponent stem cells, because they have the potential to give rise to not only embryonic cell types such as the epiblast, but also extra embryonic cell type, placenta cells, trophoblast cells, and the hypoblast cells. So these three cell types are the main cell type that you would typically find in early human embryos, such as the blastocyst. And later on, Carlos, one of my graduate students, joined the project and then further improved the protocol. So we initially published in 2021, and with this two-step protocols, basically treat the naive human proponent stem cells first with hypoblast differentiation media, bias these cells towards generating actual embryonic endoderm type of cells. And then we switch culture conditions, let the remaining undifferentiated cells to commit to a trophoblast lineage using different growth factors and inhibitors. And further, we improved this protocol. Initially, we reported about 15% efficiency, but now we can get up to 80 to 90% efficiency across different cell lines, across different labs. So they're very efficient. So these structures look like these. So this is a typical dish that we use to generate these structures called agrowell. Each agrowell contains about 1,200 microwells. And by placing about 20 to 30 cells, naive human proponent stem cells in each microwell, we can coax them into becoming these type of structures that eventually have a cavity and also an inner cell mass. and also an outer layer of trophaloderm-like cells, which eventually going to give rise to placenta. So this is have a closer look at these structures on the I guess on the right, from my point of view, is the human blastoid, and on the left is a human blastocyst. This is a day 5 to 6 human blastocyst. As you can see, they are very similar. They both contain this cavity and also an inner cell mass, and also we have a layer of the cells at the outside. It's called trophaloderm. So we look at the 3 lineages of the blastocyst. As I told you, there's a hypoblast cells, there is an epiblast cells, and also there's a trophaloderm cells. So we use different markers, specific markers to check whether human blastoid also express all 3 blastocyst cell type. As you can see here, the SOX2 is labeling the epiblast, and I guess the SOX17 is labeling the hypoblast, and also the GATA3 is labeling the outside the trophaloderm cells. Compared to this is a human blastocyst. Compared to a human blastocyst, blastoid indeed contain all 3 cell types that you will typically find in the human blastocyst. And we did a quantification. We compare the number of each different lineages in the human blastoid with a human blastocyst, but compared to either a 5-day, day 5 or day 6 human blastocyst. As you can see here, the human blastoid contain, each of the lineages are in between these 2 developmental stages. So they have a little bit less trophaloderm cells than the day 6 human blastocyst, and also a little bit more epiblast cells compared to day 5 or day 6 blastocyst, and very low number of hypoblasts compared to the human blastocyst. And with the culture condition developed originally by Magdalena Zarnicka-Go's lab, we can culture this human blastoid a little bit longer in a dish. So essentially, we grow this human blastoid on an uncoated dish, so they can attach. The trophoblast cells will spread, and then in the center, the epiblast cells will start to self-organize into a little bit into early post-implantation developmental stages. So this at the end of the culture, day 4, roughly correspond to about day 9 to day 10 human embryos. And here, just compare with the side-by-side culture of the human blastocyst. So human blastoid indeed can, in some of the structures, we can see this cavity formation. So these type of cavities are early signs of the amnion cavities that will later on form during this period of development. And also we can see the same type of cavity occurs in the human blastocyst. Although one thing I like to mention is that the cavity formation in the human blastoid culture in this format is very low. So roughly about 1% of these structures will have this cavity. And part of it is because it's culture condition for growing human blastocysts or blastoid into a post-implantation stages are not very efficient for the human embryos or human blastoid to self-organize properly in these conditions. And also at the edge of the outgrowth, we can see the human CGB positive cells are starting to secrete, produce the pregnancy hormones that typically you will detect using a pregnancy test. And indeed, compared to the human blastocysts, they show similar expression pattern at the edge of the colonies, and as the trophoblast cells will starting to produce this hormone. And we look at the single cell transcriptome by single cell RNA. So here, just showing this is an integrated analysis. We pulled together 7 different data sets that people previously published on the human embryos at different stages. And also in our current study, most recent study, we included our own reference data sets using day 5 or day 6 human blastocyst generated on a different sequencing platform, 10x instead of SMART-C2. As you can see here on the right, so these are the cells that are coming from human blastocyst cells, pre-implantation human embryos, and on this side is a cell type that corresponding to post-implantation human development, including one of the data sets from Carnegie stage 7, CS7 stage human embryos, and also some of the embryo models that are corresponding to post-implantation human development. So here just to shows these cells representing post-implantation human embryonic cells, when these cells here representing blastocyst cell types. And as you can see here, our blastoid are largely you're generating cells that are corresponding to pre-implantation human blastocysts. We calculated about 95% of them are overlapped with the pre-implantation human embryos. But sometimes we see this off-targeting cell types that are corresponding to a little bit later stages. We believe these are derived from when you collect the embryos and blastoid, some of the cells from other wells will stuck onto them. So that led to the noise observed in our system. So by having this model, the next we want to know is whether we can use this blastoid system to understand human development. So one of the aspects we look at it is the signaling. So so here using a JES, which is a chemiluminescence Western system, we check several different signaling pathways their status in the human blastoid. Here shows the red bar, and also the aggregate that is not looking like a human blastocyst. We call it the non-blastoid aggregate. And also we use the human blastocyst as a control. As you can see from this graph, a couple of signalings, including AKT and mTOR, are highly activated in the human blastocyst. And also we see the same trend is in the human blastoid, especially when we compare human blastoid to non-blastoid aggregate generating from the same experiment. We see the enrichment of the AKT and mTOR pathways that are in this human blastoid, but not less so in the non-blastoid aggregates. But AMPK signaling pathway, we didn't see any difference. But in addition, we performed the proteomics analysis. We actually find that the AMPK are differentially regulated between blastoid and non-blastoid. So we decided to use a chemical inhibitors to perturb this during the blastoid generation to see whether these signaling pathways are indeed in the derivation of the human blastoid. So here, just the bar graph shows that this is a control. The orange bar shows the efficiency of generating cavities in the human blastoid, about 80%, as I presented earlier. So it's about 80% efficiency to derive human blastoid. But with a different concentration of this AMPK signaling pathway inhibitors, we found that the higher concentration we see a reduced cavity formation or blastoid formation after the treatment, and also with the AKT inhibitors, pathway inhibitors, and rapamycin, which is mTOR inhibitor, we can suppress the blastoid formation as well, really suggesting that these three pathways are potentially involved in the blastocyst development. Although we need to verify eventually using a real human blastocyst, but at least this model using blastoids suggests that pathways are potentially involved in early pre-implantation human development. And here just shows that after each different inhibitor treatment, mostly the trophectoderm cells are affected, but the epiblast cells and also the hypoblast cells there's a minimum changes among these different treatment groups. But the trophectoderm cells, as you can see, the cell number is greatly reduced, suggesting that these three pathways are involved in the trophoblast development, differentiation from naive pluripotent stem cells. And also we established a co-culture system by plating human blastoid or blastocyst onto this endometrial stroma cells. So endometrial stroma cells is a cell type that the trophoblast cells will interact with intimately after human embryos, human blastocysts implant into the uterus. And we saw that by co-culturing these two different cells together, then we can have a model to model this maternal and fetal interactions using human blastoid. So these are the images that we took about 5 days of co-culture human. On the top is a human blastoid, and at the bottom is a human blastocyst growing on top of this stroma cells, endometrial stroma cells. So one thing we immediately noticed is that at the edge of the outgrowth, when the trophoblast cells making contact with the endometrial stroma cells, the cells, the trophoblast cells become multinucleated, become syncytialized, which is one of the differentiation of the trophoblast cells into this syncytial trophoblast. And so this suggests that indeed the stroma cells promote syncytialization of the trophoblast cells, especially at the outer edge. Maybe cell-cell contact are needed for this to happen. So next we examined these. We use a control which is growing this blastoid on top of the fibronectin extracellular matrix instead of co-cultured with the stroma cells, or we collect the conditioned media from the stroma cells to see whether some of the effects we observe are due to secretive factors or due to contact with stroma. And also we use the stroma cells. As you can see here, seems like the stroma cells promote the proliferation of, in this case, this GATA3 positive trophoblast cells. So there's a pro-proliferation effect of the stroma cells, likely due to secretive factors, because we also observed this using a conditioned media from the stroma. The number of GATA3 positive cells are more either in the conditioned media or co-culture on top of the stroma cells. And we also saw the same effect with the human blastocysts, although statistically it's not significant because, as you can imagine, human blastocysts, when you collect them, there are different stages. Their development is slightly varied. But with the blastoid, they're very synchronized in a way. And also, confirming our observation for the syncytialization, we indeed observe by co-culturing blastoid on top of the stroma cells that promote syncytialization as evidenced by increased cell number with multinucleus. But this effect was not reproduced just using the conditioned media, really suggesting that this cell-cell contact with the stroma cells are likely important for syncytialization to happen in the trophoblast cells of the blastoid. And we also performed the single scRNA-seq analysis of the co-cultures, and either in one of the group we use blastoid and blastocysts grow just a fibronectin dish, and in other conditions we collect the co-cultures, human blastoid and blastocysts with the stroma, and try to understand what's the effect in more details, what the effect of the stroma cells on the blastoid. As you can see from this graph, we can pretty much identify all three lineages from the blastocyst. And so this group, two clusters representing the epiblast, and this small cluster here representing the hypoblast. And these, there were several clusters here associated with the trophoderm or trophoblast. And here, this large group of cells are the stroma cells that we use. And so among those trophoblast group, we can roughly distinguish them by different gene expressions into three main clusters. So one of them is that we call it pre-MTB, pre-migratory trophoblasts. So these are the trophoblast cells that are eventually will become atropitous trophoblasts, and these cells are these clusters are more likely a pre-syncytial trophoblast, because during the processing for the single scRNA-seq analysis, multinucleated syncytial trophoblasts will have to be filtered out because of the size, and we ended up with a group of cells that are about to become syncytial trophoblasts. So they retain some of the gene signatures of the syncytial trophoblasts. And also we have several clusters, subclusters that are more corresponding to the cytotrophoblasts, which is progenitor for both the syncytial trophoblasts and atropitous trophoblasts. And by co-culturing blastoid or blastocysts the stroma, we noticed that cluster 3 and 5, in particular, during the co-culture, in this case, the blastocysts, the blue color means the blastocyst cells co-culture with the stroma, and the yellow color here means the blastocyst co-culture, just culture on the fibronectin dish. So it seems like by co-culturing with the stroma, it promotes a differentiation of the trophoblast cells into this pre-migratory cell types. And we also see to a lesser extent in the blastoid as well. And here, cluster 3 are within one of the here this group. And now we compare. Next, we compare the cluster 3 and 5, which is likely corresponding to pre-syncytial trophoblast. Yeah.
Just a quick question. How many markers are defining these clusters here? These are pretty different cell types.
So we have a data set that people publish that they distinguish all these different subclusters of the trophoblast. We use, we use the top 20 of those markers to help us to annotate these different clusters in these.
And then just a follow-up question, what do you think of that pink cluster by cluster 7 and cluster 0? Is that like ectoplast?
This one? This one, right?
No. Right.
I think this is also trophoblast cells. We believe it's one of the subclusters within this cellular trophoblast. Yeah. And the hypoblast cells are here, very close to ectoplast cells, suggesting here, like they're coming from the same source in inner cell mass. And we compare the cluster 3 and 5 with these three clusters that are more closely related to cellular trophoblast. We notice there's a lot of genes that are including different MMPs and fibronectins are upregulated in these clusters. These are similar to people observed in the cultured human blastocysts. So they're corresponding to migratory features of these cells, pre-migratory features of these cells, sorry. So as you can see here, cluster 3 is highly enriched in these genes. These are just a few examples, MMP10, fibronectin 1, and keratin 17. These are all the genes are enriched in this cluster, providing further evidence these clusters are similar to this group of cells we call it pre-migratory trophoblast cells. So with that, I'd like to conclude the first part of the human blastoid. So starting from a naive pluripotent stem cells, we typically use 20 to 30 starting cells, naive pluripotent stem cells cultured in this very specific culture condition called 5ILA, was originally developed in Rudolf Janisch lab in MIT. And by this two-step of differentiation, we allow these cells to grow, to specify, to lineage differentiate into this extra embryonic cell types, and then self-organize into a structure that mimic a human blastocyst, continuum cavity, inner cell mass, and outer layer trophoblast. And once we generate this type of structures, then we can use them for a variety of different downstream applications. For example, we can model early post-implantation development, at least to a stage that we can see this cavity emergence of the pre-embryonic cavity forming these structures. And I didn't talk about today, but there are other groups like Nicholas Riveron. They use this model to model early human development. Specifically, when they use the endometrial epithelial cells, which is the first layer of the cells that making contact with the trophoblast during implantation, the established implantation model can study this process. And because human blastoid can be produced in large quantity, we can perform these potentially perform these chemical and genetic screens using this model to uncover some unknown factors or pathways that involve human development. So now I'm going to switch gear, talk about something that developed in the lab most recently, hasn't yet published. So we know that human blastoid are very good at model this pre-implantation human development, maybe to some extent the peri-implantation development, the implantation itself, and early post-implantation, maybe a few days up to before they start to gastrulate. But we've been trying for a couple of years. We try to grow this human blastoid to a little bit later stage so that they can model a process called gastrulation, but we had really little luck. So human blastoid, one of the limitations I would say at this stage is they are not be able to model a little bit later stage of human development, at least not efficiently. So now, if we want to model these stages, including the gastrulation and also many morphogenic processes during this developmental period, including the form of these two cavities, yolk sac-like cavities, and also amnion cavities, it seems like blastoid is not the perfect starting model to do that. So I mentioned gastrulation is very important. It's conserved across different species, and also citing Louis Wolpert saying it is not birth, marriage, or death. but gastulation is truly the most important time in our life. So there is an urgency of modeling this process in humans, but as you can imagine, it's really difficult to do that. So the previous years, there are several different models using purely the pluripotent stem cells, the epiblast counterpart of the counterpart of the epiblast to model this process. You may heard of people using 2D or 3D gastruloid, basically let the prime pluripotent stem cell, because prime pluripotent stem cell are very close to gastrulation. So that's one of the reasons why people use these type of cells to model gastrulation. And you in these structures, you can see there's a differentiation of these pluripotent stem cells into all three primary germ layer, including ectoderm, enoderm, and the mesoderm. You can generate this type of cells, but just by looking at them, it doesn't really look like an embryo. Look like, in this case, it's a 3D, obviously regrowing 3D tissues and organs. And this one is looking like just a blob of cells with a different polarity. This is a 3D gastruloid. Although it's very powerful to model at a later stage, including early organogenesis. And there are many studies nowadays to use 3D gastruloid to generate early lineage progenitor. I think that's very powerful, but it is not able to model the stage that before that, the yolk sac and the amnion cavity, because then you need actual embryonic cell types. And also this is a model from Jianping Fu's lab. They were able to use prime cells to model amnion sac formations. And so here, this is a amnion epithelium, this is the epiblast. It's sphere-like structures, but again, they don't like a human embryo at all. And so to do that, to model this peri-gastrulation human development, we resort to a different cell type. We call it extended pluripotent stem cells. So the reason it's called extended pluripotent stem cells, although at the time when it is published, we didn't know that the naive human pluripotent stem cell can generate actual embryonic tissue. So there were two groups have been developed different culture conditions to generate this extended pluripotent stem cell, all called EPS cells. The reason they are called extended because they can not only generate epiblast cells, but also can generate actual embryonic cell types. Again, just like naive cells. But it turned out that these cells are not able to efficiently generate trophoblast cells, but they are very good in differentiating to this hypoblast cells. So here, just using a reporter line, which has a SOX2 driven a GFP and SOX17 driven a referencing protein, I guess M cherry here. So after expose these cells to what we use in the blastoid formation, the HDM, hypoblast differentiation media, we made a little bit of modification so that we can modulate the balance of these two cell types. In culture, we can see that the epiblast cells labeled with green here, because we express SOX2, are surrounded by the SOX17 positive cells. And this is just for a few days differentiation, but surprisingly, we can, using this culture, we can culture these two cell types together for many, many passages. So this is taken from passage 12. You can see in each different colonies, there's two different cell types. There's the green cells, the SOX2 positive cells, surrounded by this SOX17 positive cells. So this is in 2D. This suggests that there's some organization, there's some cell sorting between these two different cell types. In a way, it mimics the early development a little bit, because we know that SOX17 positive cells, hypoblast cells, and epiblast cells, they segregate, they separate, but they are adjacent to each other. In some cases, you will see the hypoblast cells surrounding the epiblast cells. And so based on this observation, one of my postdocs, Li-Zhong Liu here, I forgot to include his picture here, developed a 3D differentiation protocol, again, using microwells to starting with this EPS cells, extended pluripotent stem cells. So they're initially in green color, and then differentiate, give them a positive growth factor, combination growth factor, so that they can differentiate into these two different cell types, the epiblast and hypoblast. Now we remove all the factors, let them self-organize to see whether they can cooperate with each other into generating a structure that mimic a little bit later they want to stage human embryos. So this is exactly what he did. He started with a group of cells, like 50 or so, and then at day 2, these cells, the aggregate starting to grow, and day 5, we starting to see some of the structures form in these aggregates. You see a little bit cleavage here, and maybe corresponding to this cavity formation in the epiblast. And a little bit day 6, we see the a structure look like embryonic disc, and day 8 they grow even more. Up to day 11, we're starting to see this very prominent cavities, amnion-like cavities, or primary yolk-sac-like cavities, and also we see a cluster cells that are looking like an embryonic disc is starting to generate different cell types, which I'm going to show a little bit more detail later size. And they can grow into about 1 milliliter after day 11 to day 13, which is a limitation that roughly corresponding to the size of the CS8 human embryos, about day 20 or day 21. And then the efficiency, they are very efficient. We can generate these structures at about 70 to 80% of the efficiency, very uniformly generated. So here we took a SEM scanning electron microscope images of these structures. So this is one of the structures. We remove the lower part, we expose this what appears to be epiblast, embryonic disc-like tissues, and on the top part we believe is amnion-sac-like structures. And this is a structure that I took from the internet. This is a CF7 human embryos, although they look a little bit different. So they expose, I think this is from the bottom. So as you can see, although we cannot tell too much here, but as you can see, there is some certain complexity, starting just with the 50 also cells, they can generate these very complex structures. And so not only we can do that, but we can produce them in large quantity. We can produce tens of thousands of them, in one experiment. So these are just the one image that took from day 11. You can see there's hundreds, even thousands of this type of structures. So next, I'm going to walk you through each different steps and what does it mean, what type of cells that are specified. So this is from day 1 to day 4. As I mentioned, the cells are starting. When we start with the culture, they express SOX2 because they're a proponent. All of the cells express SOX2. So at day 2, after expose them briefly with some growth factors that promote the hypoblast differentiation, you will see some the appearance of the SOX17 positive cells here. So at this stage, they are still very well mixed. The SOX2 and SOX17 are still in this aggregate. They don't segregate. But starting from day 3, when we remove the factors, we're starting to see the segregation of these two cell types. The epiblast cells and the hypoblast cells starting to form different compartments within this larger aggregates. And day 4, largely all of the cells have been segregated, and then they form a very nice interface. So this epiblast cells is again hypoblast-like cells. So this on day 5, we're starting to see from the side view, we're starting to see the structure very similar to people reported in this stage of there's 2 different cavities. This is yolk-sac-like cavity. This is the amnion-sac-like cavity. So here's the image that I took from the internet. Seems, although it's not exactly the same, but you got my point. There's 2 cavity form here and here, and here is the epiblast layer, and also underneath there is a SOX17 positive cells starting to form this bilaminar disc-like structures, very similar to human embryo at this stage. And here just additional images showing. So here just what you typically observe in the microwell. You see the reason you see this orientation because the yolk sac growing a little bit bigger, so it's like a boat, right? They float, and then the smaller amnion sac part always at the top. So that's why you always see this green cells in the if you see from the top view, always in the center, and then the yolk sac-like cells will outside. But this is a side view. As you can see there, indeed the epiblast-like cells are here forming a little bit cavity-like structures in the middle, and you see the yolk sac, primary yolk sacs are more developed. You can actually see the cavity forming these type of structures. And here just a closer look. So this is the cross sections and look at the different. This is amnion's cavity-like structures, and this is yolk sac-like cavities. And here, just with a different marker OTX2 expressed in both epiblast and also the perimeter endoderm hypoblast. We can see they properly express this stuff. And E-cat herring only expressed in the epiblast, but not the hypoblast. So as you can clearly see from this image, and here just again different views of seeing this from the top view. You can see the cavity inside SOX2 positive cells surrounding this amnion sac-like cavity, and yolk sac-like cavity are between these epiblast cells and also SOX17 positive cells. And here just even more images showing this. And then these one thing I like to emphasize here is that epiblast cells are very nicely organized into the epithelial type of structures and the yolk sac surrounding it. So here's just the video, basically repeating what I'm just showing you in the previous slides. You can see this SOX17 positive cells outside, and there's epiblast cells in the middle, and you can see this E-cat herring which expressed in the epiblast cells. So primordial germ cells. So at this stage, we use a reporter. This is dual reporter, driven by two genes. One of them is PRDN1. PRDN1 is also expressed in the hypoblast cells as well. That's you see a lot of red colors here and in a hypoblast-like cell. And also T52C, also a marker for early primordial germ cells in humans. And we see some of the yellow cells here, which we believe are representing the primordial germ cell-like cells in these type of structures. And we start to see some movement in early on. These as we know in humans, we don't know much about information, but people have done studies in monkeys. They show that primordial germ cells can migrate from the amnion into the hypoblast. So we see some kind of migration here in this day 6 to day 5 to day 6 structures. As you can see here, these yellow cells are representing primordial germ cell-like cells, and they start to migrate and then from the amnion location to the hypoblast. So day 8, so that's where we start to see gastrulation. So gastrulation is characterized by this ingression of the epiblast cells into become mesoendoderm cells, endoderm cells. So mesoderm, the marker to marking the gastrulation is a T, brachyury T. In some of the structures, we can see these T cells starting to be expressed in the middle and surrounded by the SOX2 positive cells, and outside is a SOX17 positive cells that are either the yolk sac or the endoderm at this stage. Again, this is top view, and here just a additional view seeing that indeed these T cells are starting to migrate or ingress from the middle of the perimeter streak like structures ingress underneath the SOX2 positive cells and starting to become SOX17 positive cells. Any questions so far? Okay, so here's the video showing more details about this process during day 8, and we can see again this SOX17 positive cells are outside this SOX2. In this case, is yellow color here, and the purple is a T positive cell, brachyury positive cells. Again, we can see this. It seems like a trilamellar disc-like kind of structure starting to form in this type of structure in this day 8 structures. And here, just the closer look, we see SOX2 positive cells, and then yellow is a T. I'm sorry, T is a red color here, and then bottom seems like there's a SOX17 positive cells. So it's not perfect, but it seems like they're organized into this trilamellar disc-like structures in epiblast here in the day 8. So culturing this structure a little bit more on day 9, the structure grow bigger. Obviously, the embryo as faces growth and proliferation, and just by looking at these structures, we can clearly distinguish this bigger cavities are more like a primary yolk sac, and these small cavity here are likely to be amniotic cavity, and there's a dense cellular structure in the middle is likely to be epiblast. And here just a cross section of one of the structures, we can indeed see the yolk sac-like cavities here are much bigger. Amnion's cavity here is smaller, but marked by this epiblast cells, which is SOX2 positive cells, and CDX2 here marking the amnion epithelium. So the strongly suggest the amnion specification differentiation is complete in these structures. And when we grow this structure a little bit more to day 10 or 11, we're starting to see something interesting. As the SOX2 positive cells at this stage, they become neuroplate or neural structures. And then here in some of the structure, we can see this groove-like structure, maybe neural groove at this stage. And at the posterior end of the structure, we see cells that are co-express T and SOX2, which is a population called AMP, neuromesoderm progenitors, which is going to give rise to pre-somite mesoderm and generate somite structure in humans. And we do see some of the AMP type of cells here at the posterior end. So here just another example cross-section about day 10 and 11. We can clearly see this epiblast or neural plate-like structures and marked by SOX2. From this image, it looks like they're forming a tube, but we're not sure. Maybe just a density of cells. But this at the anterior end, there is a neural plate-like structures, and the posterior end, you can see that's where the gastrulation start to happen. So they indeed establish this anterior posterior axis. And here, just another image showing the SOX2 positive cells starting to form tube-like structures and then co-express PAC6, and PAC6 cells are starting to migrate out. I mean, their cells outside the SOX2 positive become PAC6, suggesting more differentiation in the neural lineage. As I mentioned earlier, pre-somite mesoderm cells will eventually give rise to somite, and during somatogenesis, there is a very characteristic cyclation of some genes called segmentation clause, for example, Has7. So this is a mouse embryo. So Has7, they use Has7 reporter, GFP reporter. As you can see, while the somite is formed, these genes undergo this cyclic oscillation of the expression. And in our model, it doesn't look that good, but we can see this is Has7 reporter. We can see Has7 positive cells in these structures. And when we record it does seems to start the oscillation at this stage. So we're continuing to improve to see whether we can see the real somite structure forming in these structures. Excuse me. So we'll perform a single scRNA-seq analysis of these day 11 or advanced. We call it the peri-gastruloid because they can model this peri-gastrulation development in humans, for lack of a better name. So in this peri-gastruloid, we see a variety of different advanced cell types that roughly corresponding to all these different three primordial germ layer lineages, including the ectoderm, mesoderm, mesoderm. We see a lot of cardiac mesoderm cells in these structures. And also, very interestingly, we see also a group of blood cells, which I'm going to mention in more detail. So here, we generated about 12 different clusters, 13 different clusters, I believe. And each different clusters are annotated based on their unique gene signatures. And we perform an integrated analysis, integrate our dataset with published human embryo datasets at this stage, and also published monkey embryo dataset at this stage. So a couple of weeks ago, there's a dataset on human PCW week 3 human embryos, which is roughly correspond to Carnegie CS10 stage, a little bit later than our structures. But we do see there's a very nice correlations. For example, the red color here is our data set, and the blue color here on this side is the human embryo data set at week 3. We see largely their overlap in different cluster, but we do see some of the differences. For example, here we see the human embryo data set have cells largely separated from our data set. We believe that can be attributable to the different developmental stages. Indeed, when we use the monkey embryo data set, this is one of the data sets we generated earlier last year. So these data sets corresponding to Carnegie stage 8 in the monkey embryos, and in comparison, we see more overlap of our single cell RNA data set from peri-gastruloid with this monkey data set, suggesting stage is this, because the difference observed here is likely due to different developmental stages. And based on these, the reference data set from human and non-human primate, we annotated many of our clusters. And there is one very interesting cluster that we observed is this blood or endothelial-like clusters, because we know that the first wave of hematopoiesis happening in the yolk sac, primary yolk sac, and we were thinking that maybe in our advanced peri-gastruloid, we start to see this first blood cell population occurs in humans. And here, just we perform immunostaining using one of the markers called CD34. We could observe CD34 positive cells in this case, day 12, day 11, day 11 to day 12 structures. Yes, that's it. So with that, I'd like to thank all the people that are involved in this project, especially Yulei Wei and Le Qian Yu, the two postdocs that are pioneered the human blastoid work, but they have left the lab and started their own lab since last year. And now Carlos is the leading person that taking care of all the human blastoid project in the lab. And Li Zhong, sorry, I didn't post up his picture today. He's a postdoc in my lab, essentially single-handedly developed all these peri-gastruloid protocols. And I'd like to thank all the funding sources for these type of project, because I didn't, the NIH as money is not allowed to be used on these human blastoid or even peri-gastruloid projects. So luckily we have funding source for NICEF and also ASRM can support this work. And also these are my whole lab, about 14 people now. And I'd like to thank you for your attention. If you have any questions, I'll be happy to address it.